The vast majority of patients are African American because the mutation was brought over during the Middle Passage, during the slave trade, because all the evidence suggests that the mutation arose in Africa 10 to 20,000 years ago. Why did it arise in Africa? Well, this mutation would never have spread, but it's spread because if you carry one copy of this mutation, you are more likely to survive an attack of malaria. So in a region or a country or a continent where malaria is endemic, having a single copy of the sickle cell gene is actually a positive thing for your health and for your longevity and your ability to reach adulthood and have children and pass on the gene. But then the more often that you pass on the gene and other people do the same, of course, then that increases the chances of two carriers having a child. And sickle cell is a recessive disease. So two carriers, each carrying the gene, have a 1 in 4 chance of having a child with sickle cell disease. Decades ago, scientists realized, clinicians realized that if they could just switch on another form of hemoglobin, the gene is sitting in our DNA, but it's been shut down since shortly after birth. If we could reawaken this form of fetal hemoglobin, that should, in principle, compensate for an adult hemoglobin. So that's been the goal. CRISPR finally provided the gene editing mechanism. About 15 years ago, scientists conclusively found the weak link in the pathway that would allow fetal hemoglobin to switch back on. One of the biotech companies I mentioned that launched in the Boston area shortly after the Doudna-Charpentier papers was the company called CRISPR Therapeutics. This was the company that Charpentier co-founded with some good friends. And they decided that their first shot on goal to get CRISPR to work in a human being in the clinic would be in sickle cell disease and the related disease, thalassemia.